So I think what I'd better try and do is not to give you the history of molecular biology, which you all know, but to try and tell you why I think we've got to change our entire approach to medicine and why it'll all be rewritten in the future. And I think this is also very important from the point of view of society, because so much of our society will depend on how we actually begin to view human beings. So I want to tell you something which you all know about, and which is the seat of all of our troubles. We are living with genomes that ceased biological evolution, say, 100,000 years ago. So we've got a 100,000-year-old genome inside of us. Right. So, good, all right. So, how do we cope with this? What does it mean? Now, let me give you one example. Times changed. When, let us say, if you were still the slave of biological evolution and the weather got cold, the ice age was upon us, only those who could regrow fur or hair to keep themselves warm, only they survived. But man is different from all other animals. What he did is he went out and killed an animal and put on its skin. And he kept himself warm. And that's technology. That's a new dimension in the world. He did not have to depend totally on the limitations of his genome. Of course, in order to guarantee his survival as an animal, which we were before we became human, our whole metabolism and everything we did was dominated by our genomes. And what the genomes did is they specified in our hypothalamus, which is the most important part of the brain, that exactly how we survive. And what did the hypothalamus tell us? It told us, eat as much as you can when you get food, convert it into fat, because fat is the best way to store energy. All right? And so when the famine comes, you will be able to survive. And of course, that is still within us. And of course, it still dominates our behavior. But of course, now in Southern California, the famine never comes. And so instead of the survival of the fittest, we have the survival of the fattest. And I mention this because, as you all know, the obesity epidemic is upon us, right? And of course, we have to deal with this, and people don't understand it, but of course it is this 100,000-year-old genome telling us to behave in a way to guarantee our biological survival for a completely different set of conditions when we were hunters and gatherers in a very sparsely populated world. So I think we must start with this concept to really understand what everything is being produced by us has come to mean. Now, of course, we have discovered many ways of patching our metabolism. It's called the pharmaceutical companies. These are just repair jobs. And of course, what everybody would want to know is, what is the fundamental change that we can produce? And I'll come back to that at the end of my talk. But I want you to understand that our problems of humanity are the problems given by a maladaptation of what we are as biological organisms and the environment that we have created. We create this environment, and we now find our bodies are maladapted to it. So I think that that's very important to understand right at the beginning. Well, how are we then to view human beings as to be able to study them, understand, because there are still immense problems of understanding how we start off as a single cell and then land up with this incredibly complex structure that we have with dozens of systems of regulation, and of course this brain that we have, which is capable of all of what we do as human beings. And we would like still to understand this. Now, I wrote many years ago that we must learn how to convert data into knowledge. But only very recently I've come to understand, I've come to add yet another step. I think we need to convert knowledge to understanding. And I think the best example of the difference between those is seen in the animals of the world. All right, so there are thousands of animals, different animals in the world. That's data. Explorers found them. They're still finding new species of insects. They're still discovering them. It was Linnaeus who converted this into knowledge by making a classification. But to understand all this about all these different animals, we had to wait for genetics and Darwin. We had to wait for evolution, the concept of evolution, and we had to wait for the rise of genetics. So this is still what is lacking. And even if we have knowledge, so-called knowledge in medicine and in human biology, we still do not understand how all the systems work. And I think that that has become the biggest challenge of all that we have to master. Now, I, of course, don't like this word translational medicine, okay, because I think it's the wrong approach. The idea behind translation goes roughly as follows. The extremely bright people called basic scientists, they work in labs, they work on mice and flies and worms. They're very clever people. But we need some way to translate their findings into applications. So we need to bridge this distance between the basic science, the basic biological sciences, and the applications to medicine and even other parts of biology. And that's the wrong approach, because I think it is always characterized from bench to bedside. It is wrong, because the problems of humanity, the biological problems of humanity, are discovered in the bed. Right? And what we must bring to that is the science that will convert this into understanding, so that we need to go from bedside to bench. We need to add science. And I think that in the future, I'm prepared to make an absolute prediction that we will tackle directly the problems of humanity by putting science right into the hospital. It's happening already in the world, but until we get most of it directly attached to the study of people, then we will still lack. We will have this idea that we do it on mice and then we apply it to man. And if I had my way, I would declare a moratorium on mice. Later we can study mice. The big problem about mice is they have no money to pay for drugs, so we don't have to cure them for that. All right, so now doing that means, now why has this been so? I think the most important step was taken in the mid-1980s when it was decided that what we would do is to sequence the human genome. I mean, that was, in fact, a massive program which wasn't thought of. You have to realize that at the time these discussions took place, the only organism that had its complete genome done, the biggest one, was the bacteriophage lambda, which is 45 kilobases, 45,000 bases. That was the biggest one. And then the talk was we would jump from this and go straight to humans with 3,000 million bases. Okay. And of course, this did require a huge development, and it was carried out. Now, it was likened at the time to sending a man to the moon. It cost about the same. It was estimated to cost $3 billion. You realize with the money people talking about now, that was trivial. It was peanuts. 3 billion is nothing. You know, when banks go and they need 200 billion to be rescued, 3 billion is just, you know, a few pennies in the game. So, but it was a big project, and of course it was worldwide, took a lot of difficulties, a lot of problems to get it going, but it was done. But in a sense, it was exactly like sending a man to the moon, because, you know, sending a man to the moon is easy. It's bringing him back that's difficult. Bringing back means it could be quite easy to do it. And so I want you to think of this genome is stranded on a metaphorical moon by itself, and what we knew would be the big difficulty is to interpret the genome, to understand what is written there. And I think that I just will tell you a little about the sort of things I've been doing, because I decided about 3 years ago that nobody knew what was in the genome. Of course, computers knew about it, but computers are incredibly stupid. They are obedient, but stupid. And so everybody was doing whole genome assays and doing statistics on everything, but nobody, so I decided I would read the human genome. I would start at the left end of chromosome 1, and I would finish when I got to the right end of the Y chromosome. And I've been doing this, and I am quite amazed what is inside of it. Now, I think that when you look at a genome and you say to yourself, what can I learn from this? Well, first of all, you always have a feeling it must contain information from the past. There should be still, if at all possible, some record of what it was like in previous occasions. And I think that that is indeed a challenging problem, but I think it is doable. That is, to reconstruct past genomes from the evidence in the human genome. I choose the human. We could choose any one of the existing genomes, but the human is the best sequenced as yet, and it also turns out that we are amongst the slowest evolving of animals. We're evolving much more slowly than most of the other mammals. So far, I think I've only found one evolving slower than us. It's the elephant. But it's hard to get hold of elephants, as you see. So I think that, that we have to do that. Now, people may say, well, this is something that's of no value at all. It's of no value at all. But I think there's the basic biological science is embedded in these genomes in two ways. One is a record of the past, and we can prove that the human genome does contain information from the past because it's not at equilibrium. So there is a whole analysis of genome structure that we can do that, in fact, once you get into this, you can start to see which are the parts that have changed least and which are the parts that have changed most. And this in itself is very interesting. We can also see the main reason for complexity is that the genome, there are multiple copies of the genes, and they have specialized in different ways. And in fact, it could be argued that to make a genome, to make an organism as complex as ours, we can't do it with one, or even we have to have multiple copies. At some point we have to duplicate the genes and then evolve different ones. And this you can see in the genome. For example, if you tackle a very interesting question, which is how does the retina work? You can see that the parts of it that are copies of each other, that is, the rods and cones are copies of each other. Okay, once upon a time, long ago, that is before there were vertebrates, there was only one kind of visual detector. And I don't know what you would call it. Maybe we should call it a cod or a roan, but we don't know what to call it. But it was a visual detector because we can see all the machinery has been duplicated and specialized. And we can now tell which came first in this. And of course, if you go on with this, you will find that, in fact, there's a close relation between our sense of smell and our detection of vision. In fact, the very first thing must have been in some animal that didn't look like anything that we look like now, must have been to differentiate between the chemical detection of smell and the chemical detection, because the machinery is the same thereafter. So I think we can reconstruct exactly in many ways how things are developed simply by trying to understand how the genome itself has preserved this record. Now, I think this is a challenging intellectual problem, and you won't find many people working on this. But I think it is going to be the problem for young people. And I think, and let me tell you why young people are good at this. It's because they are ignorant. I'm no good at science anymore. I know too much. So I don't try anything. But if you have ignorance, you will try. So I think you should use your ignorance. It's very important not to know too much, because then you won't try to do anything new. So I think young people need to go into these fields, because I think the revelations there will be as great as the revelations that we had in the last century concerning the origin of the universe. I think this is going to be a very important understanding of the biological universe, the biocosmos, if we can call it that, and to develop biocosmology, which is essentially evolution, and understand that completely. But of course, there's another thing that we need to understand is how does all of this function? And of course, you realize that there is a great new subject, which I think is a complete waste of time, called systems biology, which claims to reach this understanding of how everything works. I've called systems biology the following: low input, high throughput, no output biology. And it's more than a joke, you see, because if the systems biologists, I don't mind them collecting data, that's fine, but it's the way they think about the data that is wrong, all right? And what you have to say is, can you deduce from the behavior of a system the internal workings? So just imagine a man is playing a drum inside another room, and we are listening to it with lots of microphones and so on, and we're trying to reconstruct from the sounds the physical properties of the drum, how big it is, how it vibrates, and so on. It turns out that's a classical inverse problem, which in this case is insoluble, because information has been lost in the passage over there. We cannot reconstruct it. So what we are good at, and that is the role of molecular biology, is to do the forward process. That is, go and find out what the drum measures, find out all of its properties, and then you can play the drum yourself. You can compute from a drum, if you know this, you can compute the sound output from this. So the forward problem is soluble, the inverse problem is not soluble. Okay? Same is true in crystallography, because information is lost. And of course, you have to add a priori knowledge. So I think that this whole business of trying to solve inverse problems, which abounds in all of modern science, which is to try and work out the properties of the system from the behavior, is fraught with difficulties. And I think can only be done under very restrictive conditions. And that's an area of theory in science that I think is very important to grasp, that there are ways for us, instead of measuring the properties, to do the forward dimension. Right? Now, of course, that's the whole basis of the molecular biology, because now we have all the genes, so can I immediately write down all the proteins made in the human? That's fantastic. I didn't have to sequence anything. I didn't have to purify anything. I can make you a list in 20 minutes of all the proteins made. That would be pretty good. There may be some variation. Now, I want to understand what all of this is represented. And so I'll come back now to this issue. So we now talk about genes. And our talk about genes is completely ridiculous. So we talk about genes for alcoholism, genes for schizophrenia, genes for homosexuality. So many years ago, I invented a special genome because I discovered every family has an Uncle Frank. So who's Uncle Frank? Uncle Frank, he smoked 2 packs of cigarettes every day from the age of 4. Okay? He drank He drank 2 bottles of Scotch whiskey every day. Okay. He had several wives, many girlfriends. Okay. And he was killed at the age of 91, driving his Ferrari for a skiing holiday. So he has the great genome that's called the Uncle Frank genome. So I said, well, I want to know, everybody's looking for bad genes. I want to know the Uncle Frank genes. How can we get those? And of course, people say, no, you got it all wrong. He didn't have this. He was just lucky. He was right at the end of the distribution, and he was lucky. But then I thought, there could be genes for luck, since there are genes for everything else. However, I just want you to realize that we have a wrong concept of genes. We use it too loosely. And I give you one other example, which is how we think of genes is wrong. So many people said, when we sequence the chimpanzee genome, and we subtract it from the human genome, there'll be 1 extra gene. Okay, it'll be the gene for language, you see? But of course, there's an alternative hypothesis, which is that chimpanzees have discovered talking gets you into trouble, so they've evolved a language suppressor gene. Okay, so they will have the extra one. Not us. Of course, if we had the extra one, it would be called a Chomsky gene. But of course, if they have the extra one, we call it the Chomsky gene, you see. So basically, I think we don't see, because language is a very complex operation. It involves, lots of changes in muscles and everything like that. We cannot even reconstruct from the knowledge of the genome exactly why our feet are different from chimpanzees' feet. Okay? If you look at a chimpanzee's foot, it's like his hands. He can actually walk on them like this. But we've modified that foot. So which are the genes? Can we find that in the genome? And so I think there are many things that are challenges about function and specification that we will have to find. Well, I come now to the last part, because I think that this is going to be very important for the future. I've told you that a lot of our physiology is run by our brains. Actually, the hypothalamus is a very important part of your brain, and it determines a lot of your behavior. All your emotions are located in this is the old brain, the oldest part of the brain. Okay, so now, of course, we have to ask ourselves, how can we modify that? So, of course, people have looked for drugs that you can modify appetite and chemicals like this. But in fact, what we know is that the prefrontal cortex, this big part of the human brain, sends inhibitory stimuli back down to the midbrain. These are mostly in inhibition. Okay, so this means you can modify your behavior by exerting this unique part of the human brain, the prefrontal cortex. And I'm telling you, the technology to modify the prefrontal cortex exists. It's called education. I think you can talk to people. And I think our problems of the future are going to be into a much better understanding of the innate internal animals within us and how we can actually humanize them. So I think that education is going to be a very important part for the future in the world. Now, as all of you know, many societies are in deep trouble. You read every day in the papers that everybody is going to need looking after. Alzheimer's disease is on the increase. You see, it's called Alzheimer's disease, but it isn't a disease. It's just that the body runs down. It stops working. Right? It's one of the causes of it. You cannot live forever. I mean, it's quite remarkable we live for so long, that we have this long life that we do have. But of course, that everybody, and we should begin to realize that this is the result of not dying earlier. The reason why Alzheimer's disease is high now and has become significant is because we have conquered most of infectious diseases. We've conquered most of the diseases that kill people, like hunger. We haven't had a major wars, at least in the West, for a long time. So people are surviving to the end now. And of course, they're going to be increasing numbers. So I think that to understand this, and I believe it is a strong social responsibility of the people to say, well, this is our society, this is humanity. It consists of all of these people. Perhaps 30% will need special care at the end of their lives, and we must just reorganize society to take care of this. And I don't think we can shrug it off and say to people, well, you know, let medical research carry on and try and do it and try to sort out the hospital. We have to recast it because society has changed tremendously. And it's very interesting because these changes can be seen going with great rapidity. 30% of the children in London have asthma, 30%. It's very high. Okay. Now, in Singapore, which has converted from a very primitive economy and now has a big urban center, it is essentially one city, the incidence of childhood asthma is rising. It will soon approach 30%. Now, what is the reason for this? Is there some kind of antigen that abounds in cities that gives this? Or is the so-called hygiene hypothesis that we now live much cleaner lives, so we don't get immunized, we get sensitized to these antigens when we see them? So here is a problem in immunology, which is, I mean, it creates a very serious medical condition, is a consequence of living in this big experiment we've done, living inside a city. This is what you get. So we need to study all of these things. We need to understand them. So I think that the last point I want to make is the following. We need to systematize very soon all the concept that we have of the molecular goings-on inside of us. That is, we can now produce all of these things, and we can also understand all the feedback and all the pathways. I think we can do that in humans. We can at least do it in human cells. I think, for example, it would be, I believe that what we are going to do now, over the next few years, is to create completely new human models for human disease. So I think we're going to do that because we have found ways of converting cells in vitro into other cells, right? And if that is the case, we should be willing, as is beginning, just seeing the beginnings, to take human cells, take pieces of skin from people, convert them through various pathways into specific neurons, or into specific blood cells, or into specific muscle cells, and then study them. And we can do that also by studying the human beings in the different genetic compositions of them. So I believe that in the future, there's only going to be one subject. It's going to be called human biology. I think it's going to be the human sciences. And in fact, many years ago, I wrote a curriculum for an undergraduate degree in the human sciences, in which you would study man and his works. That is everything we've done for the last 10,000 years on this planet, all our theories, all our economies, society, anthropology, right down to the molecular biology. We should understand that integrated thing. And I think every politician needs to take that course and pass with at least upper division of the second class. Otherwise, he cannot stand for election. That's the knowledge we're going to need about humanity for the future. So I leave you with this, and I also leave you with the exciting thing that the molecular biology of man is going to give us discoveries that we haven't yet thought about. And so I urge everybody, get rid of the mice, flies, worms, even C. elegans. Day is past, it's obsolete. Get on to the study of human beings. Thank you.